One of the core missions of AACST, all computer science teachers, has always been to uplift and promote computational science, not just as a subject, but as a way of thinking. Since our inception, we have taken meaningful steps successfully in the direction through the initiatives in computational physics. And today it is a moment of great satisfaction to see that vision expanding as computational biology becomes a reality in our journey. And what makes this moment more special is all of you. Though the participants are joining one by one, but I would like to announce that we have 258 participants joining us today from 20 states across India, representing nearly 65 cities and towns inclusively and coming from diverse academic background, computer science, life sciences, bioinformatics, mathematics, physics, research, academia, and industry. This participation itself reflects the true spirit of computational biology, where boundaries
हाँ
disappear
माँ
and discipline begin to connect. And to guide
दुकान है माँ
this
मेरा लेक्चर चल
meaningful
रहा है मैं कर दूंगा
academic
बाद में
journey,
करता हूँ
it
बाय
is my proud privilege to welcome our chairperson, Dr. Mahesh Kulharia, Professor and Head, Department of Bioinformatics, Central University of Himachal Pradesh. Dr. Kularia is a distinguished academician and researcher who has contributed significantly in the field of bioinformatics and computational biology. His work continues to inspire in interdisciplinary learning and research. Sir, your presence today adds great value and direction to this session. We are truly honored to have you with us. Rather than me speaking more about today's learning, I would now request the most appropriate person to set the academic tone. So, sir, may I request you to kindly introduce the resource person and formally open the session. Over to you, Dr. Mahesh.
thank you ma'am thank you for this great opportunity to interact with so many great speakers and such august audience here and it is indeed a privilege now for me to introduce our distinguished resource person Dr. Sandhya Prashant Parasnath Dubey. I am so sorry. uh, Assistant Professor in Department of Data Science and Computer Applications at Manipal Institute of Technology at Manipal Academy of Higher Education. Dr. Dubey holds a PhD in high performance computing for protein structure prediction, which is also a very important field and also close to my heart. She has a strong academic foundation in computer science with her bachelor and master's degree from RTM Nagpur University and a master's of engineering from Satyabhama University, Chennai. With over a decade of teaching and research experience, Dr. Dubey has developed expertise at convergence of computational biology, bioinformatics, machine learning, and high performance computing. Her research focuses on protein structure prediction, drug discovery, computational genomics, and large scale data analysis. She has published several research papers in reputed international journals, including the Journal of Biomedical Research, Biomedical Informatics, and Advances in Bioinformatics, and has contributed to the development of machine learning based predictive models for protein analysis and also novel frameworks for protein structure prediction. Dr. Dubey has successfully completed a research project on protein structure prediction for drug design supported by Google Cloud COVID 19 research credits. This demonstrates her ability to leverage advanced computational infrastructure. She is currently associated with an interdisciplinary project in healthcare technology, which reflects her engagement in translational and applied research. Her research interests also extend to genomics associated crop improvement and climate resilient agriculture, which is also something that we are working on, showcasing the broader societal impact of her work. She has experience in handling high performance computing environments, big data analytics, and interdisciplinary collaborations, making her contributions highly relevant in today's data driven scientific landscape. She is an inspiring educator and a researcher. Dr. Dubey is committed to mentoring students, fostering innovation, and also bridging the gap between various fields like computer science and life sciences in this case. We are indeed very honored to have Dr. Sandhya Dubey as our resource person for this seminar, and I'm looking forward to her talk. And so I invite all of you to listen to her and let's gain from her experiences. Thank you. Over to Dr. Sandhya Dubey now for her talk.
Thank you, Professor Mahesh, for your kind introduction. You have, I, you have described the complete details, but I am just starting the applications. So, thank you so much. I would find it's very interesting and helpful if we can collaborate for some good DST project. And again, the calling project for ANRF. Same thing I will tell for the complete panelists who all are there through such type of webinar and workshop. This provide us a good collaboration platform where from the different part of country we can come together and write DST or the big grants. Thank you so much. With this, I'll start the webinar where I'm going to speak on the computational biology, exploring the emerging frontiers. And the main focus will be here, exploring the convergence of biology, data, and computational intelligence. As Abha ma'am mentioned, the audience come from different background. I have covered all these three from the fundamental sides. So we can gain at the end of this presentation how we can design a research problem statement or how we can collaborate to work on it. What type of different research problem statement exist in biology where we come from different background. Few from the biological science or the basic science, the computer scientist from the data analysis or data scientist and computational intelligence and the people who come from the mathematical modeling statistic. We all three need to come together to address the coming day problem statements. So you can see here, traditionally, the biologists were the experimental scientists where they used to perform the experiment in different labs. And they used to observe those results. Based on those results, they used to provide different insight or the principles. One of the such great insight was the DNA structures, which we got from the lab. But with completion of Human Genome Project, which started in 1990 and took almost 13 years to complete, in 2003, where the objective was to sequence the complete human genome. And on completion, the project was such a huge international collaboration from different scientists from the different countries where they came together to sequence the complete genome was celebrated at the White House. This project was resulted into almost 3 billion base pair. And from there, they came to know that the human genomes between the two person, almost 99.9% they are identical. But the 0.1% difference between these sequence lead to so much difference across us. So that's it. The completion or this project was resulted into explosion of data where with this next generation sequencing or advancement in different sequencing technique, we came up with huge amount of data, which was difficult to handle manually. There come the role of data scientist or computational intelligence. The flow of today's webinar will be such that initially I will start with how the evolution of biology in data era. So where or what are the problem statements are there in the biology which we need to address from the scientist from computer science or as a person from the mathematics department. Then what is mean of computational biology or the bioinformatics? Then briefly, I will discuss about what different type of biological data are there on which we can work. the basic of central dogma of molecular biology, which is the key principle for each of these things. Then I'll discuss few of the biological database from where we can get these data to use for our project. Then the role of data science and computational intelligence. Then I'll take two problem statement, sequence alignment and evolutionary analysis. And second one, I'll take protein structure prediction. And then I'll take one example, AlphaFold, which is developed by Professor David Baker. He is a computer scientist, and 2025 he is awarded with the Nobel Prize for Biology. And then application and the future scope. So let's start the study. So as I mentioned earlier, now or today, biology is no more about the experimental science. It also involves the huge amount of data from day to day experiment. It is resulting into terabytes of data, and we require the methods to store these data such that significantly with small amount of time, you are able to access it, extract the meaningful information, and use it for different applications. So I'll conclude as today, biology is no longer just about experiment. It is about the data also. And here we need the collaboration of different scientists from the different stream to come together to address the key problem statement using which we want to improve the better human life or provide the good health. The key SDG goal here is to provide the good human health. So as I mentioned, with completion of Human Genome Project, the explosion of biological data started, where single human genome contain around 3 billion base pair. This contain almost 20 to 25,000 of different genes. And sequencing technologies now generate the current day sequencing technology. In each experiment, it is able to generate terabytes of data, which is huge amount of data. And to address this, I believe in 2005, IBM has dedicated supercomputer referred as a Blue Gene. So from there, the high performance computing era came as an application for the bioinformatics or the computational biology people. We also have the multiple different type of omics data, such as genomics, proteomics, transcriptomics, and metabolomics. All this means that biology has become data intensive science, and we need a tool or different algorithm to handle it. The research gap, or why the traditional biology falls short initially when we were able to do, and where now they need the support from the technical side. Now the advancement if technology is no more only restricted to technical side, it also required in the biology. Why? So first thing is handling massive and complex data sets, which is beyond the capability of traditional biological approach, where no more scientists are working on single genome or the single molecule. Earlier, they used to perform this task on limited amount of data set, where the focus was only on the individual gene or the protein. Now, with the current time, we are dealing with the entire genome and their genome network. Challenge is not just about collecting the data which we are getting through the different experiment, but as a product, what we are getting, the result from the experiment, we need to analyze and interpret it. The huge amount of data, what we are getting, it is like raw material, and the significant or the information which we want to derive is difficult to identify manually. So how to extract meaningful insight from these data is a challenge, and there comes the role of computer scientist. How a person from computer science can help into it. So our main requirement is provide the approach for managing these large data set so that the access time will be small, even though we have huge potential high performance system. The accessing and the storing of data set is very important to perform or to get the real-time predictions. So this provides us a big research gap where we need to provide appropriate data structure and the database to store these data significantly. Then before storing, once you have designed the data structure or the database, before keeping all the raw material or the raw data, what you got from the experiment, we need to perform the cleaning and pre-processing of this data, identifying that which parameter we need to store and which we need to avoid it. After storing the data, next task is performing the statistical analysis, and from there identifying the pattern and correlations. Why we are doing this? Some of the examples are listed here as finding the gene associated with the disease. We need to find which part of gene is associated to a disease, analyzing the gene expression pattern, and based on that pattern, we are trying to predict the biological functions. To identify the biological function, we need to identify the change between the two genes which lead for a particular function. In addition to data science, where the role of mathematicians or computational intelligence come, there comes the machine learning and optimizations. Because the data set is huge, you have the method for storing or analyzing the pattern. At the same time, these problem statements are NP hard problem where we need to use the different optimization algorithm and machine learning approach to find the patterns. For example, for protein structure prediction, if you're going for the ab initio prediction and following a basic HP model or hydrophobic polar model in predicting the protein structure or folding it, we need to or we model the problem statement as minimization problem. So where we will find that the maximum the hydrophobic amino acids are together, lower the energy, better the structure. So all these problem statement we need to design or model into optimization problem statement and use the different machine learning techniques to predict. Classify the DNA sequence. It comes under what? We need to identify with different sequence similarity or alignment. We need to identify the different mutations and anomaly, model the complex biological systems. For instance, optimization algorithms are used in protein folding. Deep learning models are used in the genomics. These are the two basic examples I have considered. There are many more which you can explore. One of the recent or the best example what I can list here is genome sequencing of COVID-19 virus. The sequence where the scientists have sequenced the virus genome to track the mutations and analyze the different variant, and based on which they came up with the vaccinations. Since the computational support was there, it was easy or it took less amount of time to sequence the genome and come up with the medicines. The next application of this is personalized medicines, where you will not treat a person based on the condition. Using the machine learning technique, we can predict or tailor the treatment based on individual genetic profile. Still, it is going to take long amount of time to go for the individual genetic profiling. There involve multiple ethicals and other parameters, but one of the objective is also to go for the personalized medications, keeping the individual genetic profile private or secure. So this is only possible because of large scale data analysis and AI, because as I mentioned, in every human cell, there are 3 billion base pair, which result into almost 20 to 25,000 of genes. Analyzing it is not easy. We require the assistance from the AI. So biology today is not just about the experiment. It is also about the data computation, and intelligent algorithm. All these three are the three different stem or the branch where we can perform the different type of research. And with this, I can state that modern biology is driven by data and solved by algorithms. So there come a role of us to come together. I request here all the people who are from the biology, please come together with the people who are from the computer science and discuss the problem statements. If we work together, we will able to address the problem statement at the earliest, and we will able to help each other, because again in the computer science core domain the research is very difficult or very limited, where we will be working on the architecture, how to improve the performance and all. But with current advancement in the architecture, we have enough potential to perform the computation, but we want the problem statement from the biology, where we can make use of all the advancement which has taken into the computer science to provide as a solution for the biologist or the scientist from the other basic science department. So from there comes the term as bioinformaticians. What it exactly look, it is a merging of the people who come from the computer science, biology, and the statistics. If we all three come together, we will be able to address any problem. So we'll become the Superman to address any problem statement. In brief, why we need the bioinformaticians, which is the combination of all these three fields: biology, computer scientists, and the statisticians or the people from the mathematical department, to analyze and interpret this biological data. Bioinformatics is science of extracting the meaningful information from the data what we are generating in the biological lab or wet lab. Earlier, it was done independently only, either in the collaborations of people from the biological lab, physics people were supporting, all the basic science people were there. But the next pillar is of the science is now computer science, where we are standing as providing the algorithms or the techniques to support the scientists who are working on this life science project. These are some area which is listed why we need. So we need to store the biological data. These are the three key parameters which I listed prior also. We need a significant or accurate methodology to store the data so that access will be fast, analyze it efficiently, and derive the biological insights. So now I'll come to the second part of presentation, the biological data type. The three different type of data sets are available in the biology. One is DNA, RNA, and protein. From the principle, it is stated that all the genetic information is stored into DNA. So it refers as a blueprint of life, where you are coding the complete human life using the 4 alphabets ATGC. ATGC get converted into RNA, which act as a messenger or intermediate molecule to convert the DNA information into functional form. And this will convert it into a protein. So protein is a final functional molecule. Now, protein is consisting of 20 different amino acids, which are from ATGC. The three codons, they will form the codons of 3 letters, and each will regulate one or different proteins or different amino acids. And these proteins are considered as a working unit of cell. So from this amino acid sequence, we have protein structure, and protein structure regulate the cell functions. So if you want to do the research, these 3 different type of data is available. We can concentrate on any one, or you can also concentrate in 1 or 2 together. So central dogma was proposed by this scientist, where he has provided the DNA structure, where he say that information flow from DNA to RNA and RNA to proteins. So DNA is mainly considered as information storage. Information transfer is done by RNA and functional execution is taken care by the protein. Any mutation in DNA can affect the RNA and ultimately the protein structure and functions, because if there is a change in the DNA or mutation in the DNA, it will affect the RNA, and the change in that particular codon will lead to the change in different amino acid, which will change the folding, and folding will change the structure, which will lead to misfunctioning or wrong functioning of the protein, which lead to the disease. Hence, that is the reason why we need to analyze the DNA data. That is the core component. Now, after we know that these are the three different type of data using which we can perform the different experiment or different research, from where you can get the data. These I have listed here few of the database from where we can get these biological informations. This database is used to organize these biological information such that it allow easy access, search and retrieval. So primary database contain raw experimental data. Examples are GeneBank, EMBL, and in detail also I'll go through this database. Then we have secondary database which contain curated and interpreted data. Example is PFAM and ProSite. Then specialized database, which are focusing on the biological aspects, specific biological aspects only we will see. So why GeneBank, or what is the GeneBank? GeneBank store the DNA and RNA sequence. If you want to work on the DNA or RNA data, you can get the data from the GeneBank. It is maintained by NCBI and which contain million of sequence. Any sequence which come into the lab, it need to be deposited to this particular gene bank. Next, if you want to work on the protein data set, you can use the UniProt protein database, which store the protein sequence and functional information. This also include the protein structure, functions, and domain. And next is protein data bank, which store the 3D structure of protein, which is important from drug design and the structural biology. Database are the backbone of bioinformatics because they provide the data on which all analysis is provided. Since they don't, scientists or the panel was not wanted to limit the access of data to only to the lab because of huge amount of fund put on the Human Genome Project. All these data sets are available online. Excuse me. So for instance, if you want to access the DNA or RNA informations, you can use this website. It is from NCBI to access the genetic information. It also provides the different tools which are come up in different lab. For example, BLAST, Intercell, Zine, and more. And using this tool, you can perform different type of analysis. It provides the sequence analysis, all the different sequence analysis algorithm or sequence and alignment algorithms. And next one is EMBL EBI. It is from the European Molecular Biological Laboratory. You can access it from this particular link. I have shared the PPT, and from there you can access the link. This is also known as ENA database. Similarly, we have DDBJ from Japan. So this is managed by National Institute of Genetics from Japan. Similar part of genetic information, DNA and RNA, but it is from the Asian counterpart. INSDSC, this is International Nucleotide Sequence Database Collaborations. These three databases are synchronized daily as part of INDC. So whatever new gene we get, we need to deposit to this database, including or to ensure there is no duplication of the effort. no other lab, if one particular gene has been already identified, no need to put the additional effort on identifying the same thing. Global data sharing and consistency across all these database. So this is the brief, the different database and their type and purpose is mentioned here. If you want to work on the nucleotide, these are the database from where you can get the DNA or the RNA sequence. If you want to work on protein, UniProt or PIR you can use. And if you want to work on protein structure prediction, you can use PDB, SCOPE, or the CAT database. Now, unlike primary database, which store the raw sequence, we also have the secondary database, which contain analyzed, curated, and biological interpreted informations. And we can make use of those available informations. They are created by the expert or the software pipeline by identifying or analyzing the raw data to identify the pattern, domain, motif, and functional annotations of these data sets. Two examples I have listed here. One is PFAM, which is the database of protein family and domain. Whenever we come across a new protein, we need to identify or map it. It belongs to which protein family. And this can be done using the PFAM database, where it provides you multiple sequence alignment for each family. It uses HMM to search the new sequence. It provides the functional annotations. The next one is ProSite. It is again a database for protein family and functional sites. This is mainly used to provide the conserved motif and the functional sites, patterns and profile for detecting these motifs in the new sequence. Way to use it, it helps in predict the functions of unknown protein based on the conserved motif. It will try to map or align the sequence, and based on the conserved region, it will tell that what is the conserved motif. Why it is important? Because we need to identify the conserved functional region, predict the protein function from the sequence, and support comparative genomics and evolutionary analysis, where we will see how the evolution took place. The file format used in this database or data sets are mainly FASTA for nucleotide and protein sequence, where you will get the protein informations with the PDB ID and the complete sequence, what is the length, how many sequences are there. Similarly, you have the PDB for the molecular structure. These are the few tools which are online available, which we can make use, and based on the working principle of these tools, we can provide a new algorithm or as a result, new tool to perform similar tasks with a significantly improved performance. NCBI BLAST is used to find the homologous sequence. In this, we compare the two sequence, either two sequence alignment or multiple sequence alignment, where we compare 3 or more sequence and identify or map the sequence for identity, similarity, changes, and all. And based on those changes, we'll try to find out the key points. What is the difference which may lead to the different functionality of the sequence? Cluster omega is mainly dedicated for the multiple sequence alignment, and PFAM to identify the protein family and domain. Based on the similarity, it will map the protein to the existing family or the domain. So how, as a data science, we view the bioinformatics? It starts, I'll not say it is replacing the traditional method. No, we are supporting or coming in coalition to reduce the time to perform or to take or come to a conclusion point. So sample is the first key point, which is still done in the lab, where we are getting these genetic informations from the blood and tissues or the cell in the lab. Using the different sequencing technique, we are going to get this biological material or biological details into the digital data. So from sequencing technique, we will get the digital data. there come the role of data scientist or the person from the computational science to handle this huge amount of data and come up with appropriate algorithm for sequence alignment, machine learning algorithm to find the different pattern or the correlation between the sequence. What is the benefit or why we are doing this? To identify the different diseases, drug target, and learn or understand the evolutionary relationship. Raw biological data is useless unless we process it. And algorithm is the key to convert this raw data into meaningful knowledge. The objective of complete this thing is we are having a large amount of data and it is difficult to analyze manually or get the meaningful knowledge. So using the help of AI and computational technique, extract the meaningful knowledge as early as possible. So bioinformatics integrate biology and computation. Is a summary. Biology data come in multiple forms. We need to identify on what type of data we are working, whether we are working on the DNA or RNA or on protein. Key for all these three different type of data is central dogma of biology, which say that genetic information flow from DNA to RNA and RNA to protein, and protein is the key molecule which regulate the functionality of the cell. Now we need the different database, data scientists to convert this data into useful insights. As I mentioned, I'll be addressing two main problem statements. One is sequence alignment and second one protein structure prediction. So quickly, I'll go to the sequence alignment first. So comparing sequence alignment is nothing but comparing the biological sequence to identify the similarity and the difference. You can perform this either for the DNA or for the RNA or for the protein sequence. And based on the sequence similarities or sequence alignment, we will try to get the how much the sequence is identical, what is the difference, or how now the mutation has taken place, or is there any insertion or deletion between the sequence with the time. Why do we do this? To identify functional similarity, detect the mutations, and understand the evolutionary relationship. If two sequences are similar, it often means that they have similar function or common origin. As in the wet lab, the protein structure of prediction took place either using the NMR or X-ray crystallography, which is both time consuming and laborious task. So to reduce that, instead of going for the protein, we'll try to map the DNA sequence or RNA sequence, and from there we'll see what is the difference or how the mutation has taken place. A simple example I have taken here for pairwise alignment. We have two DNA sequence, ATGC, TAC, and second sequence. We need to align it 1 to 1 and need to see how they are similar. So if the amino acids match exactly, I'll show the tool on BLAST how it works. So if wherever the sequence are same, for instance, AA is matching TT, CG is different. So there we have the two parameters, whether they are similar or not similar. If they're strongly similar, we have the different indicator. If it is strong, weak similarity or different nature, we have the weak similarity. And based on that, we get the similarity score. There are different metrics proposed to perform or to calculate the similarity score. For example, like blossom or farm. We also try to introduce the gap between the sequence alignment to get to maximize the similarity. The second method is multiple sequence alignment, where more than two sequences will be aligned to find the conservation or to perform the phylogenetic analysis so that we will able to identify from the origin of the species how the divergence has taken place. For example, human DNA versus chimpanzee DNA, when comparison has taken place, it was found that 90 to 99% the gene is similar. Still, we are able to find so much difference between the two species. This high similarity indicate that we are close evolutionary, we have a close evolutionary relationship and common ancestry. Even a small difference in the sequence can lead to significant biological difference. This brings us to connection between the sequence alignment and evolution. So if two species have high similarity sequence, they evolve from, we consider that, or we have the hypothesis such that the evolution is from recent common ancestor. These are some of the applications why we need to perform the sequence analysis to build the evolutionary tree, to study the species divergence, how the mutation has taken place and from where the species diverged, trace the virus mutations. The best example is COVID variant. Tracking was done using the sequence alignment. Sequence alignment compare the biological sequence and help in identifying the similarity and functions. The two main algorithms used for the sequence alignment is Needleman-Wunsch algorithm and Smith-Waterman algorithm. One is for the local alignment and second one is for the global alignment. When we are aligning it, we also need appropriate scoring and gap penalty guide to quantify the sequence alignment. So that also provide a good domain or the problem statement on what basis we are providing the scoring matrix. So sequence similarity is a window into evolutionary history using which we can see how the evolution took place. So second problem statement, already 718, I'll go through fast. It is protein structure, motif, and structure predictions. So proteins are the molecule that actually perform the biological functions. For example, enzymatic reactions, transport, and cell signaling, and their function is directly determined by their 3D structure. So protein structure prediction is one of the problem which need to be addressed. Till date, for available protein sequence, hardly 1 to 2% of the structures are solved through the lab which are available, and using which we are using those solved structure through the computational method to predict the structure. And based on those predicted structure, we will try to identify or check it in the wet lab whether the structure is correct or not. And accordingly, it is deposited on the PDB database. So protein structure is mainly classified into 4 groups. First one is the primary structure. Primary structure is nothing but the sequence what we get from the DNA to RNA and RNA to protein, which is mainly consist of 20 different amino acids. Secondary structure, these protein structures are mainly consist of alpha helix and beta sheets based on the amino acid property, and their interactions together. They are mainly grouped into alpha helix and beta sheet, regulating the different functionalities. So in secondary structure prediction problem, we have the Chow-Fasman algorithm and others, using which we are able to predict whether the set of amino acid will lead to helix or beta sheet. And if helix, whether it is 3x3 or larger helix. So that is also one of the existing great problem statement to be addressed using the ML. Next is tertiary structure. Tertiary structure is overall 3D shape we are trying to get, which will consist of alpha helix, beta sheet, different turns and coil. So these are mainly based on the principle or how we are modeling the 3D structures, is considering the hydrophobic interactions, disulfide bonds, and ionic reactions of the amino acids. So the core structure prediction is based on hydrophobic. If you ask me the basic problem statement from the tertiary protein structure prediction, it is proposed in long back by Dill with the name hydrophobic polar model, where he has considered only the hydrophobic interactions of the amino acids for the globular protein, where he has stated the problem statement as a minimization problem statement, where we need to convert the protein sequence into hydrophobic or polar molecule and fold the sequence such that all the hydrophobic amino acid will form the core and polar amino acid will surround it. Lower the energy for every adjacent hydrophobic amino acid which are not adjacent in the sequence but in the folding, it will lead to or consider for the energy computation. Lower the energy because the scoring was, it was model as a minimization problem. Lower the energy value, better the structure. Then quaternary structure, where we'll have the multiple protein chain and combine together, and we'll see how the structures are. For example, hemoglobin, I'll try to see show the structure on the protein data bank. So since the data structure determine the functions, even a small structural change can affect the functions. So motif in proteins, the next part is, and other than the structure predictions, the motif are conserved structures or the sequence pattern that have specific functions. So in the motif finding, we are trying to identify the enzyme active sites, DNA binding regions, or the zinc finger motifs based on the applications. Motifs are like functional building blocks, and they often conserve across the species. If there is a change, it may lead to a particular malfunctioning of the protein. So protein structure prediction mainly comes under two: homology modeling, where we are going to identify the similar sequence, and hypothesis is if the sequence is similar, it may have the similar structure. So we need to find the known proteins, perform the sequence alignment and find the closest sequence, considering that as a template, and model the structure based on the existing solved structure. But the issue is we know only 1% of the protein structure. So finding the similar sequence is very difficult. Second one is ab initio method. Ab initio method is predicting the structure from scratch, considering the physiochemical property of amino acids, where it is considered as or define the problem statement as energy minimization or folding simulation problem statement. Issue what we are facing in this is computationally expensive and huge number of possible structure. Even though with the basic model, the protein structure prediction problem comes under the NP hard problem. So if you go with the traditional algorithm, it will take the more than the space time. So we need to use the optimization technique. There is no method. By considering all the structure, we will come to know how to solve the structure. Instead of that, we are using the different optimization techniques such as genetic algorithm, particle swarm optimizations, or different live-based evolutionary technique or optimizations algorithm to predict the structure randomly. Initially, we randomly design the structure, and performing the different tasks of these algorithms, we will come across the final or the optimal structure. AlphaFold is a big breakthrough of where we have where the use of AI and the computer science was done to predict the protein structure. This is mainly based on the template-based modeling actually. And this is developed by the DeepMind to predict the 3D structure directly from the sequence. No sequence alignment or the gap between the sequence and structure is tried to be filled by with this particular server. What it do, it take the amino acid sequence, use the deep learning techniques to learn the pattern and predict the distance and angle between the residues to build the 3D structure. It has achieved nearly experimental accuracy and solved the long-standing problem folding. That was the main reason why Professor David Baker was provided with the Nobel Prize, last year Nobel Prize, even though he come from the computer science background. Now he is completely or purely in the computational biology, where he is concentrating on the development of different server to model the protein structures. So AI has transformed the protein structure prediction from decade-long challenge into solving the problems. With this, I'll conclude. I think 7:25. I was moving fast because I wanted to complete by 7:30. So I can conclude as biology has transformed from experimental science to data-driven discipline. Bioinformatics integrate biology, computations, and statistics. Large-scale data, biological data require advanced algorithm and AI techniques. The sequence analysis and protein structure predictions are key understanding for biological process, and AI breakthrough like AlphaFold are revolutionizing the biological research, which will be crucial in healthcare, drug designing, genome analysis. Future biology is algorithm driven, opening a new frontier in the science. I hope I was able to justify to take your 1 hour of time. Thank you so much.
Thank you, Dr. Sandhya Dubey, for wonderful session.
Thank you, ma'am.
Now the floor is open for question and answer session. So participants, please put your questions on chat, if any.
It was a very remarkable lecture, which covered
Thank you, sir.
the entire set of basics, which of course is saying a lot because it covered a very wide range of subjects. And you know, I'm myself very happy with this lecture because this covers the basics and fundamentals which everybody should learn.
Yes.
So thank you, Dr.
Thank you,
Dubey.
sir.
And
Thank you, sir.
I'll take my privilege back if that's okay, Dr. Abha.
Yes.
And let me invite the questions from the audience.
Yes. The audience
Should ask questions.
please put your questions.
Start the ball rolling. It's always advisable.
Anindita?
Can I ask the first question, if that's okay?
Sure, sir. Yes, sir. I'll try to answer from my knowledge,
Of course.
whatever I have.
No, ma'am, of course, you have so much of knowledge, and your basics are fantastically sound. Uh,
Thank you, sir.
I was thinking of solving the protein structure by AlphaFold and so on. Many large language models and so these methods are very nice. They are indeed very nice. But perhaps it is more important that nowadays we should look at solving the interactions. I'm sure some steps have been taken. I would be very grateful if you could, enlighten the audience with these steps that have been taken to solve the interaction between protein, or protein RNA or RNA and other molecules, because that's perhaps where the entire modern drug designing is focused on.
Yes, sir. Protein interaction is one of the key challenge for the drug designing, how we are able to find the motive or how they are interacting. So there are different server or different tools are available to perform the protein interactions. But I have not worked much on protein interactions. I have worked on protein interaction in the perspective of secondary structure predictions, how the secondary structure will lead to the protein interactions. And there we have used the LSTM model to identify these interactions. Based on the
Thank you, ma'am.
Chop-Passman algorithm, we have used the LSTM to model the protein interactions.
Yes, ma'am. This is great. Thank you.
Thank you, sir.
Excuse me, ma'am. There is one more question from Krishna Jaiswal, I think. Yes. How we put the research data on the computer as in digital format? Like, could you please explain what's the process of integration of research findings on the computer?
A research finding on computers means you are telling depositing to the database. Is it with respect to depositing your lab data or lab informations on this available database? Okay. So see, if you go
Yes.
through if you go through this database, now they have mentioned a specific template. You need to provide the details on those template, and you can deposit the data on the particular database. So consider if you're working on the protein structure, you need to deposit the data on the PDB. So there exists a PDB format. We need to send the details in that form, and you need to first sign up, register login, and you can provide the data set there. The complete details are there. Actually, I'm not purely from the biological side. I have just used the PDB database by modeling the algorithm. So I use that. I'm the user of the data set. So I have explored these things little only. I don't know in detail how to deposit the data. I hope I was able to answer, Krishna.
Yes, ma'am. She says, thank you so much, ma'am. Dear attendees, you can ask your doubt in the chat box.
See, you can see here, this is...
Yes, ma'am. Vaishali Uke is there. Hello, ma'am. Vaishali this side. I have a question. There are two methods for protein modeling, AlphaFold 2 and homology modeling.
Yes, ma'am.
can you please tell me which is the best for her PhD research work?
Ma'am, homology modeling is having its own limitation. The reason is homology modeling is depending upon the salt structure which is already existing. And as I mentioned, till date, only 2% of reported protein structures are solved. So there is a large gap or exponential big gap between the salt structure and the existing sequence. So you may not able to get the similar sequence, because until you get the similar sequence, you're not able to predict the structure. So that is the main region or limitation with the homology modeling. When you go with the ab initio method, you do not require any template or the similar sequence. We are directly modeling the structure or folding the protein based on the amino acid property. But again, it requires a huge amount of computational power. And the different principle you need to understand the amino acid properties, the physical principle, what is involving, or the mathematical modeling, and using which you can predict the structure. So I will mention, or I will tell you, decide based on the facilities which is available at your research center and how you need to move ahead. But again, at the same time also, even though there is a limitations, there are these servers which are also based on the homology modeling. So first they will go for the homology modeling. They will try to find the similar sequence. If it is not existing, try to fold it based on the ab initio approach. So integrate the both instead of working on independent methods. I hope I was able to answer.
Yes, ma'am. Yes.
Thank you, Vaishali ma'am.
Ma'am, Dr. Pranil Jagdale have a question. He have DNA cocci sequences of insects and want to publish a phylogenetic research paper.
Phylogenetic tree. Huh?
Yes, sorry for my pronunciation.
Huh.
From where should he start, and which tools and methods are recommended? It is
So for-
possible. Is it
it's
possible to work in collab?
yes, ma'am. Yes, ma'am. There are the tools which are already there exist in the phylogenetic tree constructions, where again it is based on the sequence similarity. You need to find the sequence similarity, and then you need to trace this complete evolutionary tree. So if you just Google or explore the online resources, sir, you will able to get how to perform the phylogenetic analysis. Actually, I mentioned initially I will include the genetic evolution also, but due to constraint of time, I understood I'll not able to complete it. So I do not included that part into the today's talk. But it is one of the best applications of the biological data to understand or learn the evolutionary relationship between the two sequence or two species through the phylogenetic tree. And there are tools, sir. I have not explored the tool. Actually, my research is restricted only to the protein structure prediction. Any other questions?
Yeah, well, I would encourage the audience to ask as many questions as possible.
Yes.
This is the opportunity they won't get
Thank you, Pranil,
repeatedly.
Dr. Pranil.
I think we should move forward. Or you have any questions? Shall I wait?
Perhaps it is Saturday night and people want to go
Yeah,
back.
yeah. So correct, sir.
Yes.
Thank you.
Ma'am, there's one more question from Krishna Jaiswal.
Mhm.
Ma'am, is it possible to get PPT of today's webinar? Right.
Yes, we will put in group.
Okay, one more question.
PDF of this presentation will be shared in our WhatsApp group. Okay.
Okay. Ma'am, one last question. Ma'am, are you doing any research on agricultural crops?
Yes, ma'am. We have submitted a proposal for Badigi chili. We want to modify the crop to increase the production. So a small pilot work we have completed. We have submitted the proposal and it is under the review. But as such, no such dedicated lab I have. We are working on small scale. So we are waiting for the fund.
Thank you, Dr. Sandhya, for engaging interaction. To bring everything together with perspective and insight, I now invite our chairperson, Dr. Mahesh Kularia, to share his remarks.
Thank you, ma'am. This lecture was a wonderful lecture, and it helped, I'm sure, all of the audiences to gain the complete perspective, a bird's eye view on various fields that are there in the bioinformatics and various databases that are available, tools that they can make use of. It was indeed a very nice lecture, and I'm sure, you know, we could have gained with more publicity of the lecture. Perhaps if more students would have joined it, it would have been very beneficial. And Dr. Sandhya Dubey is doing phenomenal work in research, and you know, more people should join bioinformatics. It's not a very difficult field, as we have seen. Dr. Sandhya Dubey is a person of computer sciences, but she's doing very wonderful in bioinformatics. So it's an open field, and I would also encourage the audience to take an example from this lecture, and should focus more on bioinformatics. And I was also very heartened with the number of questions that we had gained. Although, of course, as a teacher in me is always looking for more questions and so on. But and it was a wonderful organization step by Dr. Abha Khandelwal, and I am very grateful that you have created this platform where such events can be organized and with such quality. So I sincerely, I'm grateful, and more of such events must be organized. So, in fact, I would recommend that in future, if you are doing such kind of webinars, faculty in my department can also join in, and they can also be part of this process. And there are more such groups in North. There's another group in North in Bhatinda, Central University of Punjab, Bhatinda. They're also doing computational sciences, and they're doing wonderful. So perhaps at some point of time we should also include them also. And with these words, and with a best wishes message to everybody who's doing in research. We do research only for our satisfaction. So all of you will gain satisfaction if you continue doing your research. So all the best to your research, and may all of your projects become fruitful, and you gain a lot of experiences and create wonderful things. And maybe, and I hope that perhaps in our audiences, we will have people who will achieve Nobel Prize at some point of time. So with this, I would say thank you, and over to you, Ma'am Abha.
Thank you, sir, for your valuable insights and encouraging words. Now, I would like to call Anandita whether she would like to take photograph before vote of thanks or after vote of thanks. Just let me know. Okay, she must be busy, so let us move.
Yes, ma'am. We can take, ma'am, now.
Now we can take.
Yes,
okay,
yes.
so please. Rupa, come on camera. Priyanka, come on camera. Priyanka.
Yeah, people should switch on their video feeds. This is the time to be happy about. The lecture is over.
It's a memory.
It's a memory indeed. Yes.
And then you can see on website, no? Event gallery, you can visit.
Done, ma'am.
Done?
Thank you all.
Thank you.
Thank you, Abha ma'am. Thank you, Mahesh sir. Thank you, Nandita ma'am. Thank you, participants, for your
Thank you,
precious
madam.
time.
Wait. Let Rupa play her role first, then you leave. So as we gently move towards the conclusion of today's session, I invite Ms. Rupa Rajkumari, Assistant Professor, Department of Computer Science, Hislop College, Nagpur, the same college where I had served as head of the Department of Computer Science for 27 years. And when I got retired, she entered. So I'm inviting her to propose the vote of thanks. Over to you.
Thank you, ma'am. So it is indeed my privilege to propose the vote of thanks on this occasion. I'd like to begin by expressing my heartfelt gratitude to our founder, mentor, and convener, a special mentor to me, Dr. Abha Khandelwal. Ma'am, I've had a privilege to working with you since the beginning of the AACST. And I always closely observed your work. It is always thoughtful, focused, and result oriented. And today we saw the result, ma'am. And you have a unique ability to think ahead and turn your ideas into reality. Your creativity, thinking, and dedication are truly inspiring for all of us. And thank you once again, ma'am, for giving all of us this platform and for constantly guiding all of us. And my sincere thanks to our esteemed resource person, Dr. Sandhya Dubey, ma'am, for delivering such an insightful and enriching session. Ma'am, your expertise and clarity have truly added great value to all of us. I really hope many researchers are going to take this field in their research. Ma'am, I also extend my heartfelt gratitude to our respected chairperson, Dr. Mahesh Kulharia, for his gracious presence and for sharing his valuable insight that enriched today's session. My special thanks to our coordinator, Dr. Praveen Gosekar, Secretary of AACST, for his continuous support in orchestrating all the arrangements, right from preparing the flyer to managing registration and certificates seamlessly. I would like to sincerely thank our technical host, Dr. Anindita Kundu, professor at SP Raisoni Women's College, for ensuring the smooth conduction of this webinar. My appreciation to the organizing committee member, Dr. Prajakta Sharma, and Dr. Shaba Nazim for the dedicated efforts behind the scenes. A special word of thanks to our friends, Professor Aravind Kulkarni and Dr. G. Mahesh Kumar, for being a part of this event. Your presence and participation added great value and warmth to the session. And finally, my heartfelt thanks to all the participants. Your presence, enthusiasm, and engagements made this session truly meaningful. Once again, thank you, one and all.
Thank you, Rupa Madam. And before we conclude, a small request, I invite Dr. Priyanka Sharma, member of organizing committee, to kindly circulate the feedback form. And I request all the, perhaps she has already circulated. I request all the participants to take a moment to fill it. Your feedback is extremely valuable to us and will also help us record your attendance. So, and with that, we close not just a session, but continue our journey, because learning like this does not end here. It grows, it connects, and it evolves. So thank you everyone for being a part of this academic vision of AACST. And especially I would really like to thank once again to our chairperson, Dr. Mahesh, and Dr. Sandhya, resource person. Have a wonderful and meaningful evening ahead, whatever is left. The session now stands formally closed. So we all can leave except the
Thank you, ma'am.
participants and
And
the
bye, ma'am.
technical host and our organizing committee member.
Bye, ma'am.
Bye, sir. Thank you. Thanks a lot, sir.
Thank you everyone for your
Thank
time.
you, Sandhya. Thanks.
Thank you.
Participants, kindly fill the feedback form and then you can leave.